from a personal view, I've just found this meeting truly inspiring as well as informative, I think. But I really had no idea how much progress n-Lorem had made in such a short period of time. And I was just listening to some of the previous presentations that the n-Lorem team was giving. And I mean, just to give you some context, in terms of INDs, 9 INDs, is that right, Stan? And so I quickly went to the Pfizer 10K and I looked at their 10K. So Pfizer has like 83,000 employees, okay? So they have somewhere over 30 INDs, but that's essentially 3 times what you've done. And this is not only Stan, this is the constellation of people around, Roseanne and Frank and everybody else who works with Stan and Amy and the rest of the team. This is an incredible achievement in the amount of time that we've seen. And I just think it's like truly inspiring to see, you know, how much progress has been made. But I wanted to just give you that kind of context for what we've seen. So in this final panel, 40 minutes or so, and I hope we'll have some time for some comments at the end, we wanted to explore how we think about expanding what's going on at n-Lorem into the research community. How do we amplify what's going on here, and how do we engage the research community with what n-Lorem is doing? And so we've got like 4 panelists for you here. I'm not actually going to introduce them myself because I want them to talk about what they're doing. But just, you know, we have chief editor of one of the premier journals. We have 2 researchers who are either at the clinical end of and at the kind of basic and clinical interface. We also have an example of how somebody who is who at some point in time, Jeff, is going to have to think about how this is going to affect you. And we also have John here, who's from one of the premier foundations that's in autism and neuroscience. So, Jeff, do you want to kind of kick things off and talk a little bit about your interaction with n-Lorem?
Sure, thanks everybody. Hi. Yeah, my name is Jeff Carroll. I think maybe the first way to introduce myself to this audience is to say that, as Sarah mentioned earlier, as well as being a professional in this field, I'm also a family member. So myself and 3 of my siblings carry the mutation which will cause inevitably Huntington's disease, a really terrible neurodegenerative disorder. Although every time I come to this meeting, I'm reminded that as terrible as HD is, seeing all these sick kids reminds me that you know, there's worse things in the world for now until we get them all fixed. So I'm a rare disease family member. I'm a scientist. I worked with Frank Bennett for many years on the Huntington's disease program, which is really the great hope of the Huntington's disease community. You know, we're deep into the clinical studies for a Huntington ASO that I think is the best hope that everybody in the HD community has ever had. And it's in clinical trials right now, and I'm very excited about it. And when that went to the clinic, I sort of thought, well, you know, what do I do now with, you know, my exposure to ASOs? And I run a mouse lab, and I know some genetics. And I met Frank after he had just, and with Stan, kicked off this amazing organization. And Frank said, hey, you know, a little bit about ASOs and a little bit about genetics and some basic science. You seem like the perfect person to rope into helping vet cases. And so I started doing that. So all those 100 and whatever cases, many of those are work that I've done, and now with lots of help from other people because we're way past Jeff's capacity. So I help vet the cases, and so I'm kind of on that early part of the funnel, and it's just been incredibly fun, rewarding, interesting in a weird scientific way, but also motivated by the fact that each of these is just a really sick family. And so just incredibly motivating and fun, and it's been a great honor to be playing that role since the beginning of n-Lorem.
Great. John?
Hi, everybody. I'm John Spiro. I'm a neuroscientist, and I work at the Simons Foundation in New York. It's a private nonprofit founded by Jim and Marilyn Simons. Jim was a very influential mathematician turned investor, and instead of buying like a sports team or something, he decided to start a foundation to give away money to advance the frontiers of science. And there's been a special focus of one part of the foundation that I'm involved in called Safari on autism research. And we made a decision about 17 years ago to take a really big bet on genetics. So this was in the early days of autism research where really, other than things like Fragile X or Rett, there really wasn't much understood about the genetic architecture of autism. So we spent a lot of time putting together large cohorts of individuals with autism, and then at the time doing deep exome sequencing. Early on that was a big deal. Now this is becoming routine, and so and when I say we were funding researchers all over the world to do this research, and we basically uncovered that there's this enormous, autism, which is often considered a sort of common disorder. I think one way to look at autism, especially on the sort of more profound, serious sides, is really a collection of rare disorders. And so we've upped the ante, so to speak, in the last few years and created a cohort called SPARK that aims to be the largest effort to collect families all over the United States. They either participate remotely online or through a series of clinical centers that we have relationships with. And we're trying to do phenotyping of these families, but also doing very significant genomics research that we've sequenced something on the order of 50,000 individuals with autism. There's something like 150,000 in this cohort that haven't been sequenced, but that are sort of involved in the research. And the idea is to sort of create this research-ready cohort. There's a lot to learn. I think there's an enormous amount that interfaces with what n-Lorem is doing. There's a lot more to say. I might stop there, but just want to say one more thing is, in addition to creating these cohorts, Safari has put an enormous amount of effort into sort of doing the behind-the-scenes analysis of what these genes are doing, where they're expressed in the brain, making iPS cells and things like that. And so we've spent a lot of money on animal models, and I still think there's an enormous benefit for animal models, what we can learn. But I think something that I've come to believe from watching n-Lorem and some other players in this space is the enormous importance, when it's safe to do so, really important caveat, the enormous importance of moving drugs into humans when it's possible, because I think we're going to sort of hit a wall with what we can learn at some point. I think other people have made that point today. But anyway, I just want to, maybe I'll stop there.
Great. So Vijay, you're kind of at the cutting edge of clinical research, kind of at the Broad. And so can you talk a little bit about your research and kind of what you're seeing and how that interfaces?
My name is Vijay Ganesh. I'm a neurologist at the Brigham and Women's Hospital, where I co-direct the neuromuscular genetics clinic, so patients either with a diagnosis or looking to obtain a diagnosis from the community in neuromuscular diseases. I run that with my division chief, Tony Amato. On the research side, I'm at the Broad Institute functioning in really two arenas. One is a project called the Rare Genomes Project, where this is a patient-initiated research study where patients apply to us who have a suspicion from their clinical team of having a rare disease, a single gene disorder. And we apply research-based advanced genomic methods, whole genome sequencing and a slew of other methods that I can get into, to get a diagnosis for that individual. So this is essentially a research study where both we're hopefully applying a service to these patients, simultaneously academically advancing what genomic methods need to do to improve the diagnostic yield across all rare disease patients. In addition to that effort, my role on the research side is to do this kind of analysis specific to neuromuscular disorders, where it's a particularly difficult problem of improving our diagnostic yield for these patients. There's many reasons for it. These are very large genes that can tolerate a lot of variation in the non-coding, the kind of silent part of the genome, where we know that causal variants exist, but how to bring our advanced methods to prove that to put it on a platter basically for n-Lorem to say, okay, this is the cause. That is all kind of, I think, where on the academic side we can really interface with efforts such as n-Lorem to basically both build the best practices on the research side and also deliver to these patients variants with the evidence from their cells, and then have a best practices approach so that these methods can be applied more broadly and expedite that process. We know it takes time, and time is essential. So broadly speaking, on how our group at the Broad, the Rare Genomes Project and some of these other consortium efforts are run by Anne O'Donnell-Luria and Heidi Rehm, my mentors. There's 3 parts to it that I think we all see as being essential, but somewhat separate parts to it. One is the genomics methods development, applying those tools. The second piece of it is redefining these disorders. A lot of these patients are telling us that our conventional ways of making these diagnoses are increasingly antiquated and might complicate matters. So that it takes a physician effort and translating it to the genetics to sort of even frankly rename these syndromes. I mean, as Neil's an expert in ALS, we still diagnose ALS, or the essential part of establishing the diagnosis is hitting someone with a stick, at their reflexes, and then sort of determining if that's abnormal or not. And we can, in what I see often are neuromuscular, muscle diseases, it's defined by what people see under a microscope. But we know that many genes can cause a similar appearance. So on the research side, it's really to spearhead that effort, to connect the genetics and what we know about it to redefine these syndromes and establish a more rigor about how we establish these rare diagnoses, to unify a lot of patients that are disparate, labeled with different things. And I think it'll help a lot of efforts that we can do on the academic side to help the patients going forward. And then the third piece that our group does, particularly Heidi Rehm and her impressive efforts to cause improved data sharing globally, is link up patients with sort of open data sharing. Nano-rare is one thing, but it really helps to identify, both for the patients, that second individual in the world with the same disorder. It helps form a human connection for these patients. I've seen this in my own rare disease discovery work, but also from the scientific side, what are the overlaps for those patients? How do we redefine that syndrome so that now that we can see it's a shared entity? That requires a global effort and requires a lot of things that I think can only be spearheaded at the academic side. So those are the things we're sort of involved in.
Great. So Bob, you're kind of even earlier than everybody else here. You're kind of seeing what's coming through basic research in terms of technologies. So if we kind of have exome sequencing as the kind of clinical standard to an extent, so what kind of technologies are coming through the pipeline at the moment?
Do you want me to introduce myself first?
Yes, go ahead.
Okay. Yeah. You might know who I am the chief editor of Nature Biotechnology, so I have taken over from Andy, which is why he knows me very well. As apart from these guys, I left the lab about 10 years ago to become an editor, and have been at Nature Biotech and leading it the last year. And so I obviously publish your research, your maybe not the clinical trials, but more of the basic science research that goes behind the clinical trials, earlier stages. So in some ways, I'm seeing the earlier stages of the research in that way. But yeah, also I'm sometimes late to the game because you guys are all doing the research, ahead of submitting your paper. But also, I have the pleasure of, I worked with Stan and Kelly on this amazing comment that they wrote earlier this summer on the, promoting n-Lorem and what they're doing here, and just speaking about ultra-rare diseases in general and what we can learn and what needs to change in the system. So as a journal editor, I get to do both the research side and also some of that commentary and bring some of that discussion and community to this field. I can get back to your point about technologies now. So in terms of technologies that we're seeing, I think there's a lot to be said here. I've listened to all of these talks today, and there are a lot of limitations that we're working through, whether it's in terms of diagnostics, identifying these mutations earlier, whether that's prenatal testing or moving from whole exome sequencing to whole genome sequencing and isoforms, which are differently spliced proteins that can contribute to disease, and putting those in more context of what is actually functionally happening in the cell. And that involves a lot of different technologies. So, it's sequencing technologies, it's protein technologies, it's, and then being able to link all this back to phenotypes is just, it's difficult, and we need methods to do that. We need a lot of data to do that. So, I think that's where all of you are coming in. So
this is a really good lead into this kind of question as to, how do we get the kind of diagnosis, genetic diagnosis, improved in terms of the return on, a positive, I have the pathogenic variants here. Clearly, we're further ahead in terms of understanding coding sequence than we are all of the regulatory sequences. And so does anybody want to kind of chip in, talk a little bit? Maybe VJ, do you want to?
I'll start it. Yeah, I'll start. This is a big problem. I mean, this is essentially if you look at diagnostic yield of genetic testing, there was a huge bump from the technology, the widespread application of exome sequencing. Basically, a lot of rare diseases can now be resolved by applying exome sequencing. Before we were doing single gene testing or even gene panels. So that's a big advance. But now it's sort of petered out in terms of our improvement in diagnostic yield. We need a technology advance. Genome sequencing was thought to be this advance, and it's reasonable to consider that in terms of the acquisition of the data that it actually is, but it's really the interpretation problem. We're probably capturing what are pathologic variants. We just can't say that they are, and that's a fundamental barrier for this that many people have described in very elegant ways. But is how to solve that problem is a combination of a data sharing issue. We need to just collect more phenotypes and more genome sequencing, and then some model over time will resolve which are pathogenic variants and not. So there's an in silico computational big data aspect to that. And then the second part of it is methods beyond the genome to try to resolve pathogenicity. And that can be RNA sequencing, protein sequencing, and methods yet to be sort of developed. There's methylation assays, things like that. But each of these kind of have to be, it's a combinatorial approach. Fundamentally, that's what we do at the Broad. And so they're not necessarily competing with each other. They can work in parallel. But fundamentally, for the nano-rare patients, we have to figure out not only which is the best method, but can we deploy at scale across the whole genomics community and return it in a reasonable amount of time. So this is unresolved, but I think that's the fundamental challenge.
Jeff, do you have some points to make on that?
Yeah, I was just thinking about from the n-Lorem specific perspective, the cases that we've gotten. Very often we're lucky enough, not lucky enough, but we're lucky enough that it's a mutation in a potassium channel in a known residue that lines the pore. And there's physiological evidence from the literature, and it's very clear that if we could get rid of that malformed ion channel, we could probably help this kid. But then we often get, and I'm sure the clinicians are very familiar with this, but a deep intronic mutation that's kind of near a splice site, and it's maybe that's causing this patient's phenotype, but there's no publications in literature, there's no functional data, and as amazing as n-Lorem is, we're not set up. We're set up, as you saw, we're set up to make 9 INDs, but that means that there aren't people at the bench doing basic research into these mechanisms. And so I think one of the struggles for us going forward is how do we build partnerships with researchers who can do those proof of concept experiments to really, because we really need to be sure about the mechanism, right? ASOs are amazing, but they can't fix every kind of genetic lesion. And we're good at this because we're careful about what cases we take on, and they're ones where we think ASOs can help. And there's often a sort of a maybe 5 or 10% gap of proof in the literature that makes it difficult to take some patients. And that's I think our future ambitions is how do we grow that and make those relationships with researchers to help close those gaps.
I feel that there's a lot more to talk about here in terms of variant analysis, but I aware of the time. I also want to kind of talk about another aspect that I know, John, that you've thought about a lot, which is kind of biospecimen kind of collection and how you use that, and how do you kind of start to use those kind of patient-derived materials. Can you talk a little bit about that and how that might apply to these kind of ultra-rare patients?
Yeah, well, one of our guiding principles at Simons has been that we want to bring really good people into the field, and we know that one of the barriers, there are a lot of really smart people out there looking for interesting problems to work on, and we know that one of the barriers is access to really access to really clean data and to biospecimens to go along with it. So, we're a private nonprofit. We don't hold any IP. We don't have any kind of financial stake in everything. But we've basically tried to, just like you were saying, for individuals who are sequenced in these really large cohorts like Spark, we've tried to take families who have identified highly penetrant mutations and help them start to form communities around some of these gene groups. And as part of that, then, because Spark is sort of a go broad but go a little bit light, so it's sort of try to find as many as people as possible, get them into this sort of pipeline, but sort of a light amount of information. But once we sort of can identify one of these highly penetrant mutations in these individuals, we enroll them in this sort of parallel project called Simon Searchlight. And the idea there is to go much deeper if we can. And part of that deeper aspect is trying to collect blood and other biospecimens. We have an emerging effort that I think is going to be really important. I think most people would agree with that, in trying to have iPS cells that are produced in a really clean, standardized program and made available to any researchers around the world, essentially for free. So we think that's really important. Something else that goes along with it, I don't think it's been mentioned that much here and maybe not emphasized that much during the day, but in this other program, we're also trying to really push hard on ways to bring quantitative longitudinal phenotyping to these cohorts. So it's terrific to bring people into the clinic and to do overnight EEGs and stuff like that. But we're really pushing hard, experimenting with different ways to bring sort of wearables and different types of gadgets into the mix as well, and get data on people that's longitudinal and that's sort of in the home setting and stuff. So I think it's a really exciting area because there's so much technology being pushed into this area that I think that's going to be really exciting.
If I could just interject, sorry, I'll try to be brief, but the 2 days before this meeting, I was across town at the group meeting of the 9th IND patient, talking about his imminent treatment, which is really exciting. And Anupam Gupta, who's another neurologist at MGH, showed data from longitudinal tracking of alternating an ankle monitor and a wrist monitor in this kid, and then applying these incredible machine learning techniques to the barrage of data, and making these really amazing symptom tracks in sort of feature space of this kid's symptoms. And sure enough, he said, it looks like this got a little bit worse here. And the parents like, oh yeah, he was doing terrible that month, and it's like it was seen in the data before they said he was not feeling well that week. And I think that's the future. And I think with rare patients, because we can deeply phenotype them and we're intervening with this very specific on-mechanism treatment, it's a really amazing chance to develop these techniques, which could then go to the broader community.
So this is really interesting to me. So we had Kelly here as one of the patient representatives, and she has some kind of amazing kind of patient-derived kind of data, yeah? But there are services like Citizen kind of coming online that are also kind of finding ways of putting EHRs together with, other types of data like that. So what do you think is the power of that technology? I mean, Jeff's just given us a really good example. Do others kind of find that really exciting?
Short answer is yes. I mean, getting electronic health records are kind of, we're living, at least in the US, in a very fractured healthcare system, and even just changing internists, like trying to figure out, how to move records over can be really challenging. But yeah, I think that's really exciting. I think there are a lot of groups and a lot of money moving into this wearable, nearable space. I think one area that's really ripe for monitoring is sleep. I think this is a really important state of the brain, especially for CNS stuff, and the technology for being able to monitor sleep structure and sleep architecture, I think, is really coming online. So I'm very excited about this.
Yeah, I am too. I'd actually emphasize some old tech aspects of it too. I mean, I drink the Kool-Aid as a neurologist that the examination is so elegant and powerful, and helping us. And it was created, a couple 100+ years ago. Still has utility, but we now can apply these sort of other phenotyping tools too. What I would add, though, just as a practical matter, of our system is just that the majority of cases that n-Lorem sees, and the majority of the rare disease patients that I see too, are neurologic. I think it's just our sensitivity to picking up on these phenotypes. There probably are ultra rare liver diseases. We just don't have a way to clinically notice them, I suppose. But the patients do kind of tell you these things, and I think a little bit of increased awareness in taking care of rare disease patients to things that are outside of your domain. Like I think Stan showed a chart of some GI symptoms in patients, and that like level of awareness, I think I myself struggle with in phenotyping these patients. And I don't think like a technological solution to that problem is necessary. It's just to get increased awareness to it, that you may be seeing the primary problem be in the central nervous system, but that actually clarity of the biology of the disease itself from an academic standpoint, and also from tracking symptoms from an outcome standpoint, biomarkers, that just like this awareness, and you're taking care of ultra rare patients, that it is multi-system in a way, it would help. And I don't have a solution to that, but I would say that is the siloed idea within medicine of phenotyping, and our each field's template for how they write their examination and notes kind of is a barrier to this integration.
Bob, any thoughts?
I mean, I don't have much to add to all of those great comments, but I will add that Jeff, you talked about how these wearables are really benefiting from the use of AI, which I think is a really point that extends past the phenotypic data, and it's the integration of that phenotypic data from these devices along with the other omics and modalities that we have. And I think AI does have a lot of potential there, to just touch back on that point, because integrating all of these modalities across a large patient set, you have multiple similar phenotypes with different gene mutations and similar gene mutations with different phenotypes, and to be able to dig more into that, the use of AI, I think, is also very exciting. So that's a little off topic anyway.
Yeah, but it's really interesting to me because nano-rare, the issue is kind of linking things together, yeah?
Yeah.
And so it seems like there's no reason not to find ways of doing this in terms of the kind of molecular data that different patients can generate, and finding ways to link those things together over international borders. But then Vijay and I were just talking last night, like a problem that kind of nano-rare has with kind of specimen research is like getting specimens, like traveling between different places is super difficult, yeah? Vijay, do you want to talk a little bit about that?
Yeah, I mean, this gets into a little bit of the system changes that have to occur in like academic medicine to basically enable this type of ultra research. I mean, it's again a bigger problem, but data sharing is helpful. I mean, so and these networks that can link different scientists, but it still requires that be the purview of that person's job to sort of follow up on it. There aren't I mean, many patients that come to us eventually kind of say a similar thing. They had a motivated physician taking care of them, but like this ultra deep dive into a rare disease is just not practical at the community level. So we have to have that sort of structure of somebody eventually up the chain has to be incentivized to do this, and then link up globally to do that. The regulatory stuff is probably beyond my scope to know how to solve it. But just even the first contact, there are tools some of you may have used, like Matchmaker Exchange. That initial contact that somebody else exists with this can, right off the bat, spearhead all the MTA and downstream things that are necessary. What you were speaking to about prospective collection of cell types and cell lines, I think is really helpful too. So that you don't, some things that slow up rare disease work is that, a line would be helpful here, and going back to the patient to do a skin biopsy to generate that can slow things down.
Yeah, it's almost like, it seems to me all of these different kind of technological solutions are nascent at the moment, and it's going to take a few years for them to kind of kick in. Like I'm thinking of also jumping back to the kind of ability to look at things like longitudinal data, like you have all of us, yeah, which is kind of looking at healthy individuals. But that's just starting to kind of get off the ground. So in the last 10 minutes or so, also, if anybody has a question from the audience, do feel free to raise your hand. Yeah, we have a question over here.
Yeah, my question is for Vijay, and especially as Medicaid has, I think in 7 states, has started covering whole exome and genome sequencing, and I'm wondering, if you might, and you touched on it a little bit, but if you could guide us in how we communicate with our community, what's the difference in turnaround time from a clinical prescription for whole exome or genome sequencing and enrolling in something like the Rare Genomes Project?
Good question. Very practical one. I don't have like an exact. I can give you a framework of that answer. It is true that I think increasingly, many people. I think we're speaking to an audience that's already converted here. That genome sequencing is ultimately kind of needs to be a clinically accessible test for some patients. Maybe reasonably not first pass, but at some point, fundamentally, the answer could be obtained without a longer odyssey, without genome sequencing. It won't solve everything, of course, but that needs to happen. So the increasing coverage for that is still a practical barrier for me. Enrollment, and so turnaround time for a clinical exome is pretty reasonable. I think 6 weeks or something, I think is what typically been my experience. On the research side, we're much slower. So the turnaround time in the Rare Genomes Project for genome sequencing, just to even run the sequencing and get the data back before we would even interpret it is right off the bat about 4 to 6 months sometimes, and then the interpretation can really take a much longer after that. And that part of that is these are already difficult, hard to solve individuals in the first place. So that's part of the bias. But also we're just not having a sort of a scalable way to turn around that faster. So in general, of course, I always tell my patients, if you can obtain this test clinically, do that first before enrolling in a research-based platform. Commercial genome sequencing is still, I think, in the space of self-pay, and the standards for what needs to be returned or not, in my opinion, are not firmly established. So it's still a world that needs a little bit more clarification over time of exactly what the deliverables are, what the turnaround times are. Places have shown you can do ultra rapid genome sequencing. Rady has shown this, that you can practically do genome sequencing in a day. But how to deploy that and do that is a real challenge. On the research side, we're much slower.
Stan.
I thought just a couple of comments. I think in the midst of the wearables and the AI enthusiasm, which I share, I think Vijay said something that may have gone by people that is a lesson that we've really learned at n-Lorem, and that is not AI, but intelligence. It's good to listen to patients and families. And I see constant examples of patients and families who try to tell professionals what's happening and are ignored. And for me, I always listened to the patient when I was still seeing patients, and that's where I learned. And in doing n-Lorem, I'm reminded constantly of the value of the parent observer for our patients, and the problem in getting physicians to actually listen to what people want to tell them. And I hear that all the time. So yeah, let's engage in AI, but how about just some simple intelligence would be a really nice step forward, I think. And the second thing I want to talk about is greed. I think we stumble over greed about 90% of the time. And we run into greed with the office in every medical center who's responsible for getting cash. And so we spend incredible hours and days and months in trying to get a simple MTA done. Yeah. And that is nothing, yes, there are some, we have to protect patient confidentiality, but most of what we deal with is greed. And it's time to get the greed out of the way and let us just help these patients, and it really annoys the hell out of me. There are a lot of things that that's just one.
You seem so mellow, Stan.
They turned on.
Oh, they'll turn it on. I'm sorry, thank you. Well, what I'd really like to ask BJ is what we can do to get Heidi and Ann to work a little more closely with UDN and n-Lorem, but I'm not going to ask that question. So
Above my pay grade, yeah.
I don't think it's actionable. You mentioned, functionalizing genetic lesions, connecting genotype and phenotype, by doing GWAS on ever larger data sets. And, we went from Nomad to UK Biobank, and we've got Estonia and all of us you mentioned. And still, what are we going to get down to? Minor allele fractions of 0.1% maybe, by going to ever larger sets. And yet severe, if I understand correctly, I'm under the impression that severe pediatric disease is largely de novo disease, and you're going to have allele fractions near zero.
Yeah.
And so I'm wondering what if you have any inspirations for what we can do to more easily functionalize those extremely rare mutations that we only see in 30 people in the world.
Yeah, good question. I think these are like the GWAS and like large data sets, all of us, genomic, these are different. I don't know, the metaphor, that this isn't my thought, but I forgot who said it, is one is like your telescope's looking into deep space, like large, vast expanses, and the other direction in the rare disease space is the microscope in that direction. You're going to see different architecture at those different scales. And so both the technology is the same how you're looking. It's just like optics for the prior metaphor, and here it's just genomic sequencing and functional experiments. But I think what you'll learn is like a better resolution of constraint. What parts of the gene are just intolerable to variation, and why is that? Like, why is that domain important? That's the academic question that I think will be resolved by that. That's very different from the population. Where the minor allele frequency is something like 0.1 or higher. That the ultra rare, where it's like unique, maybe you'll never see that particular variant ever again. But I think I think that's what the functional experimentation is going to be revealing to it, as what like high impact intolerable variation is across the genome. Yeah.
Great. So we're running towards the end of our time. I don't want to keep anybody. It's a fantastic panel. I think kind of just I suspect Stan's going to talk about this, but one other thing that I was hoping to get to that we didn't really get to was like how little NIH funding goes into this area. Yeah. And Stan, I presume you were going to be talking about that. All right. So if we could just thank our panel. Yeah.